All right, hello. My name is Jason Linville. This is being recorded for CJ 353. This last section, because I'm just a terrible professor, I forgot to put the learning objective slides, which students always demand. Please tell us our learning objectives before you start lecturing. Okay. But in this last section, what I want you to focus on is what we ended the last section with, this two-step conversion process. Okay. So we want to know how we can start out with having peaks that have assigned times to them. Okay. So this is like, what did we say? 19.5 minutes and 20.2 or what, 23 point whatever. Okay. And we want to know how we convert them into alleles, because we don't care how long it takes them. We care, is this a 7 allele, an 8, a 9, a 10? How many repeats are in those peaks? Okay. So the two-step measuring process, the first thing that we're going to do is we're going to use the size standard to convert this number time into size. Okay, so the first thing that we're going to do is say 19.5 today on this instrument, 19.5, that actually equals 186 base pairs. Okay. And now I know what you're thinking. You're thinking, oh, 186 base pairs. Well, you can just add it up and find out how many 4 base pair repeats is on there. Unfortunately, you can't. Okay, because this base pair size, it's actually an estimation. Okay, so on some days, a 7 allele may calculate, and it's not going to be a whole number. Okay, on some days, a 9 allele may be 185 base pairs. On other days, it may be 187. So that's the confusing part, because we use whole numbers, but just understand that that size, it's actually an estimate and not necessarily the true measurement. So we then use an allelic ladder that will take this size and say, okay, today on this instrument, 186 is a 9 allele. All right. So we want to go over these processes because the size standard is used a little bit differently than the allelic ladder. Okay. So the size standard, let's first focus on how do we take that time and estimate the size in base pairs? Well, if we want to estimate the size in base pairs, we're going to buy a reagent that has amplified DNA where we know the size in base pairs. Okay, this is going to be our theme here. For the allelic ladder, we're going to use a reagent with amplified DNA where we know the alleles. Okay, all right, so this size standard has known sizes in base pairs. Okay, for example, the size standard is going to contain millions of copies. So again, it's amplified DNA. It's not just thousands of copies, it's millions of copies. And I could even put in here millions of copies of labeled DNA so the instrument can see it. That are 75 base pairs, 100, 150, 200, 250, 300, 340, 350, all the way up to 500 base pairs. Okay, and actually all of our amplified DNA for STRs are probably all the ones that we work with are really under like 300, but the typical size standard will go up to like 500, okay? So any size DNA between 75 and 500, we're going to be able to estimate the size of it. All right, well, what does that look like on the instrument? How does it get in there? Well, we actually mix that size standard with our sample. So before, when we were talking about our sample as having a 9 allele and a 12 allele, at the end it has 2 peaks. Well, we're going to mix all this amplified DNA, our size standard, with it. And the end, our sample, when we run our sample, we're not just going to have 2 peaks, we're actually going to have a lot of peaks. Some of those peaks coming from the size standard and some of those peaks coming from the sample, okay? So the times of the sample peaks are going to be compared to the size of the size standard peaks. Okay, so let's go back here and let's look at this, okay? What we're looking at here is the same drawing we had before, okay? So we have this drawing up and we have our peak number 1. The green peaks are the peaks from our sample. Okay, so this, even though it's not written there, this would be like 19.5 seconds, and this would be our 23.5 or whatever it happened to be, 23.6, whatever. Those would be our time. Okay, but now what we're doing is we're not just running those by themselves. Okay, so this time no longer applies, but this actually, we're running these samples with our size standard peaks. And the way we label these peaks, the way we label this amplified DNA is we label them with different colors. Okay, so all those 10 million copies of our sample, Okay, all those 10 million copies actually have a green dye attached. So when the instrument sees it, it can say, oh, it's a green peak. And again, we're going to get to how the instrument differentiates green and orange and et cetera a little bit later. But what I want you guys to recognize is, okay, these big ones, these green ones are our sample peaks. And these orange ones, the instrument knows those are our size standard peaks. So the instrument actually looks at all of the orange peaks that come out, and it looks at, from 75 to 500, and it starts labeling them. Okay, it knows that it should see 10 peaks from 75 to 500, and it knows the first one is 75, the second one is 100, and it labels them all. So in this example, the instrument sees these 3 orange peaks, and it goes, oh, this one's orange, or excuse me, this first one's 190, this one's 200, and this one is 200. What is it there? I have a bunch of stuff in the way, 210. Okay. All right. Now, again, the instrument, because when we set up the analysis parameters, we tell the instrument we're using this size standard, so look for a 190, 200, and 210 peak. Okay, the instrument does it and it assigns it. So it knows today on this instrument, a 190 is running right here. A 200 is coming out at, like 20 seconds. A 210 is coming out at whatever time, okay? But it looks at the spacing of these orange peaks, and now all of a sudden, it can estimate our sample peak, not just in time, but it can actually estimate it in size in base pairs. Okay, I want everybody knowing that the 190 base pairs comes out here, and 200 base pairs comes out here. What size do you think this first green peak is? Go ahead and write it into the chat and let me know. Like, give me a guess. Come on. Get rid of all that ink there. Okay, we got a couple guesses for 190. Yeah, it's about halfway between, maybe a little bit less than half, but yeah, like 195, 194 is a good guess for how big that peak is. Okay, what about this peak over here? How big do you think that peak is? Give me a size in base pairs, knowing that it's somewhere between 200 and 210. Okay, yeah, we got 206, 207, 205. Great, and that's what the software does. Based on where your sample peaks are coming out compared to the size standard, which again is mixed with the sample, okay, it now can convert, the software can convert this size in time to size in base pairs. Okay, so that's how I want you guys to be able to phrase it. Okay. All right, but we still don't have the answer as far as a 194 base pairs. Is that a 9 or an 8 or a 10? Okay, 206, is that a 12, a 13, an 11? Okay, so this is the next thing that we're going to talk about. Okay, now how does the computer use that size standard? It takes those, it basically plots the size standard peaks, excuse me. So these numbers are the known size in base pairs. These are comparable to the 190, 200, 210 that we had before. And then it plots at time, how long does it take for them to come out? Okay, so this is sort of the size here, and this is the time as far as how it takes. And then it compares your sample peaks to those size standard peaks. Okay, so you can see here, this first peak is somewhere between 139 and 150. Okay, so I'm going to estimate it. It's probably like about 142. Okay, but the software estimates these peaks by drawing a size standard curve and again, estimating the size of these peaks. Okay, if it estimates that this is 195, it doesn't mean that it is 195. It means it's around 195. And in fact, what I cut off from this figure, because I don't focus it on this as much as in the undergraduate course, the estimate actually has a decimal point associated with it. So that allows you to understand that, oh, a peak cannot be, or a sample can't be 147.26 base pairs. Okay, it's either 146 or 147, but again, it's just an estimate. Okay, so the software is estimating the size, and the way you guys can visualize it is just a round number. Okay, it goes from 19.5 today on this instrument, it's a 196 base pairs. Okay, so how do we convert those base pairs to alleles? Okay, we do that by comparing our size in base pairs. Okay, again, think of it as a two-step conversion. Time to size in base pairs is done by comparing to the size standard. Base pairs to allelic ladder is done by comparing to, base pairs to alleles is done by comparing to allelic ladder. Okay, the allelic ladder is a reagent that contains amplified DNA for all possible alleles. Notice this is similar to the size standard. The size standard has amplified DNA where the sizes are known. Allelic ladder has the alleles are known. Okay. So, for example, the allelic ladder will contain millions of copies of THO1 allele 7, THO1 allele 8, THO1 allele 9, THO 10, 11, 12, 13, whatever the common alleles are, and D5 allele 7, D5 allele 8, 9, 10, 11. So the allelic ladder, depending on how many loci you're looking at, like if we were just looking at one locus, the allelic ladder would have just, like if we were just looking at THO1, it may have 8 peaks in it. 7, 8, 9, 10, 11, 12, 13, 14, and 15. Okay. If we're looking at 3 loci like we are in class, then it'll have like 8 peaks from THO1 plus 8 peaks from D5 plus 8 peaks from D13. If you're in a forensic science lab and looking at a kit with 16 loci in it, then it'll have like 8 peaks from loci 1, 8 peaks from, it has a lot of amplified DNA in that allelic ladder. Okay, but when you separate it out, all the peaks are able to separate. Okay, now the allelic ladder, again, this is where it gets confusing in comparing it to the size standard. The size standard is mixed with your sample. The allelic ladder is not mixed with your sample. The allelic ladder, we sort of run it as a control sample by itself first thing. So the allelic ladder, just like your sample, is mixed with formamide and it's mixed with size standard. Okay, so the allelic ladder is going to be run first and it's treated just like a sample. All right, so this is what our allelic ladder looks like. Again, so let's restart the day. Okay, we're trying to determine what 2 peaks are in our sample. So the first thing we do is we're going to run this allelic ladder. But allelic ladder, it's been mixed with formamide to get it into single-stranded form, and here it has these 190, 200, and 210 peaks in it. Okay. Here, there's no mystery about what the alleles are, because since we're using an allelic ladder, we know what all these alleles are. The software knows that this first green peak is a 7, and then an 8, and then a 9, and then a 10, and then 11, 12, 13. So does everybody see how the software can use this allelic ladder to set up a conversion table? Okay. Can everybody see? And this is what I want you to be able to do. If I show you a picture like this, I want you to be able to write down, estimate the size for all of these, and then make that a table like this. Okay. So we ran the allelic ladder and we have 186 equals 7, 190 equals 8, 194 equals 9, 198 equals, et cetera, et cetera. Run the allelic ladder first and you get a table. Okay. Now we run our samples. Okay. And the reason we have to do this, again, because it makes no sense. Well, wait a minute. Isn't 186 always going to be an allele 7? No, because this 186 is actually going to have a decimal point to it. It's going to be an estimate. All right. And on different labs for different days, this chart may change a little bit. Okay. It may be 185 on one day, maybe 185.4 on another day equals 7. Okay. But just know you run that allelic ladder, you get this conversion chart. All right. And then you run your sample. Okay. For our sample still has the size standard in it, right? So we run our sample and this is where we're at. Okay. We know the time. We're able to compare the time here to our size standard peaks, and we're able to estimate the size in base pairs, 194. But now that we've had that allelic ladder run, we can now take these 2 sizes and convert them into alleles. Okay, 194 is allele 9. 206 is allele 12. So even though the allelic ladder is run first as far as the beginning of the day, when I ask you guys to describe this conversion, talk about the size standard first. Okay. Say the size standard is mixed with the sample, and it allows me to convert from time to size in base pairs. And then if I show you a picture, be able to convert the size to size in base pairs. Okay. And then the size in base pairs is compared to a table that is created by the allelic ladder. And this table allows me to convert the size in base pairs to alleles. Okay. Does everybody see that? If those two peaks, if we had a 190 and a 202 peak, then the genotype would be 8 and 11. Okay, so this, again, watch this a couple times, go through this a couple times, make sure you're able to describe how you convert a peak or amplified alleles from just a time or time it takes to reach the detector into size and base pairs. And then how do you convert that size and base pairs to the correct allele? All right, and if you could do that, you can be in good shape. But also recognize there's a lot that can start to get confused in here. So if you go through this several times, you should be in good shape. All right, are there any questions about what we just covered? I'll leave the recording running for a little bit, and then I will stop it. And then you guys can ask me any question you want. I'm going to go ahead and stop the recording. And then I will continue answering questions. Again, for this class, we're going to have a quick review in spring of 2020, quick review on Wednesday, and then quiz on Friday.